Welcome to Inside The Hive TV podcast, the show that takes you into the world of bees. I'm Dr. Boncristiani. In today's episode, I speak with Dr. Olav Ruppell, a professor in the Department of Biological Sciences at the University of Albertam in Canada. We talked about his career working with social insects and also discussed his latest research publication on honeybees. The publication reveals that VARROA mites, an important pest of honeybees, feed differently depending on the life stage of the parasitized honeybee. This discovery is significant as it greatly enhances our understanding of the life cycle of this important honeybee pest, which causes significant damage to the beekeeping industry. I want to thank our Academy members for their support and you for watching. Hello everybody. Welcome to the show. It is with great pleasure that today I have my great friend, my mentor, a guy that I have a huge respect, Dr. Olav Ruppell. Welcome to the show, Olav. How are you?
Hi, Roberto. It's a pleasure to be here, and thank you for inviting me to speak on your show.
This is something supposed to be happening a long time ago, but we never had the opportunity. And I think now with this new publication that just came up a week ago, I think was the perfect fit for you to come in and share a little bit about yourself. And this incredible research that you and your team now in Canada put together is an impressive body of work. Before we jump into the presentation itself, Olav, and it's a tradition here, I ask about how you get into the bees. If you can give me a little overview about how you get into the bees and your background.
Yeah, so I grew up in Germany, and my father was a biologist. So I always knew that I wanted to do biology because he showed me what a fascinating life that could be and how good, it is to pursue your passion and play around basically and learn about life. And never stop being a child, so to speak, and asking questions. And so I wanted to be a biologist and a scientist so that I could ask questions and answer questions. Wanted to study birds, really. That's what I started university with, the desire to study birds. And then it took my mentor, Professor Hölldobler, and his animal behavior class that I took as a very fresh student to fascinate me about the world of insects, and specifically social insects. Social insects are the ants, the wasps, the bees, and the termites mostly. And Professor Hölldobler was an ant specialist. And a large part of that animal behavior class was all about ants, because for every question, there was an answer in the ants. And so he did manage to fascinate me about the social intricacies of group living and colony living. And so I started working for my master's and my PhD at the University of Würzburg on a queen-size dimorphism. So ants have, just like honeybees, a queen and many sterile workers in a colony. There are 10,000 different species of ants, and there's lots of different varieties of biodiversity in those species. I was particularly interested in ants that have different sizes of queens. And so that's how I got started with social insects. After my PhD at the University of Würzburg in the year 2000, I decided that I needed to study a model social organism. And the model social organism is the honeybee, because the honeybee is obviously really important commercially and ecologically, and a study system for a lot of different kinds of questions as well. Very well researched. And the honeybee genome was also sort of peeking over the horizon in the year 2000, even though it was only going to be published in 2006. So I thought I would learn and study the honeybee as a postdoctoral researcher. And that was at the University of Davis in California with Professor Page, Robert Page, that many of you probably know. And I stayed there for 2.5 years and got my training, got everything that I wanted. And then the plan still didn't work out to return to Germany into the ants, because in the meantime, I got married to a Canadian biologist, and we looked for 2 jobs in the world. And it happened to be North Carolina, at the University of North Carolina in Greensboro, where we could both be starting our independent research careers in biology. I found that there were so many questions about honeybees that I stayed largely with the honeybees up to this point. UNCG was our home for 17 years, and I built my career and my group there, including hosting you for a few years there for a wonderful research project. But then we were called up to the University of Alberta here in the year 2020, or I was moving in 2020. And now I have my group here where it just snowed again. So greetings to Colorado, Florida, or wherever you are. But yes, we just had a fresh layer of snow and the season is a lot shorter, but the honeybees are still a lot very productive. Great beekeeping community here up in the north. Yeah, I'm happy, very happy to be here at the University of Alberta and still researching many questions in honeybees.
And you are working hard indeed. There is several publications of you. It is in my to-do list to make videos about. So this year, probably you're going to see your group a lot of highlights of your research here in the Inside the Hive TV. And yeah, a lot of work, my work there. I have such a good time with you. I learned from you a lot about science and how to conduct yourself ethically, specifically how to do good sciences. And I want to say thank you in front of the world. You teach me a lot. Thank you.
Well, thank you. I do think, independent academics are really important because our salary doesn't depend on a research outcome or a finding. And so we really need to be that independent judge that does, the evaluation of whatever is in front of us, a question in an objective manner. And that's why tenure is so important. And I hear that Florida has just given up on that principle of tenure. And that's just a horrific.
It's scary. It is scary. Yeah, it's two different words. I think there is a book from Professor, what's his name? I forgot his name. It's a conflict of vision. That's the title. And some people, there is pros and cons in almost everything. And sometimes there is those collisions of ideas. I think Florida is in this blue and black and white, blue and green and red. There is a big conflict now. And I think we need to go back to the center. But that's my personal opinion. Olav, let me give you a little background before we jump into this new research that you just published.
Right.
Because here I have my members of the academy here with me. The academy is basically a place that I teach anyone with any background about the scientific inquiry. What is to do science in real life, the thinking method, the scientific method, all the biases, so they can be critics themselves about everything they read, is critical thinking. So that's what the academy is about. And today's subject, I think, is a great example about how science works. We have little pieces of work of specific people from different groups just adding to the story. I don't even remember how many years ago when Dr. Samuel Ramsey published in PNAS the discovery that VARROA mites feeds on fat bodies. There was a big boom. And today, I'm always skeptical when things go to the news with that kind of veracity, That creates a lot of waves that for a guy like me, a science educator now, I got a lot of trouble trying to bring people together to convince them that that's not that simple. And I even got hate emails and people pointing fingers in my face. That was a humongous, I can't even understand exactly how that works. It created a sort of cult around that subject, and we can't talk. If you say anything that's not fat bodies, done, you're done. And now you come with a phenomenal publication full of evidence with a lot of well-done experiments showing that this conversation is a little more complicated than we thought. Do you agree with my assessment?
Yeah, I think the science is a process and it's not a product, and so we have to understand that knowledge evolves over time, and in some ways the best progress is made through some controversies, because then you really question the other side. And this critical thinking that you mentioned is really important, to not just say X, Y, or Z said so, and that's why I believe that is true, but just look at the reasoning and the data. I am, in my heart, I'm an empiricist who does like to do experiments to test a hypothesis. So, typically we have a thought process or a hypothesis, and then we can say, well, if that is true, we can predict that if I tweak this variable on this side, then maybe I can see or should be seeing a change there. And if it's not true, if I find in my experiment that this is not the case, then maybe I have to rethink my theory and or my hypothesis. And so that's, I think, a good example here too, that, we have, well, in the beginning, we had sort of the common knowledge that bees are feeding on hemolymph, right? And everybody was believing that. And so that's why the Ramsey paper was so important to say, well, wait a minute, that's not the case at all times. Well, the neglect or the disconnect between the research findings and then, the public understanding was that, well, fat body feeding is important in the small part of the life cycle that the Ramsey study addressed. And that's where I think science communicators such as yourself come in and are so important to actually translate that knowledge or those raw experiments and scientific jargon into how it should be interpreted and into context. Right. And so I think our study is a little bit of that, readjusting the perspective and putting the Ramsey finding into a broader context and saying Varroa has a whole life cycle. It cycles to different stages. And that's what we need to look at if we really want to understand what Varroa is feeding on throughout its life. And so again, it's not black and white, as you say. It is probably, life is complicated and our mind works easier in simple classification and yes and no or black and white, but the life and the world doesn't work like that. And so we have to force ourselves to think in more nuances. And maybe this is true, but this could also be true in a different circumstance.
I agree. So can we take a look then? What were your findings then? Because I was not shocked. Actually, I always predicted some hemolymph was there, but not the way you showed. So I'm really, really happy to bring this to the eyes of the public now as a lesson for all of us about how science works. And there is no criticism at all about Ramsey's work. Actually, you're going to show, I'm sure, that you confirm his results, but you add another layer of data on top of that.
Yeah, that's absolutely true. And I do want to say that, this little presentation here I gave at the American Bee Research Conference that was also just held a few weeks now ago. And it is just a brief presentation giving you sort of the gist of the study. The study itself has been published and is open access, so everybody can access that study in Nature Communications. But yes, so let me share my screen here and go to presentation mode. I don't know whether you can see this slide.
Yes, we are seeing very clear. And if you can use your pointer to have some sort of marker, that would be great.
Absolutely, I can do that. Pointer options here, laser pointer. So I have my laser pointer here. And so, I want to start by saying that the real burden, most of or all of the practical work was done actually by my Chinese collaborators at the Chinese Academy of Agricultural Sciences, and specifically the Institute for Apicultural Research that you see sort of the logo of on the right-hand top corner. And I want to particularly mention Associate Professor Bin Han, who has been planning and conducting most of the studies and who is the first author of the study, and then also his mentor, Shufak Su, who is the leader of the group over there. So this is definitely a collaborative project. And you can see we start here with a slide of the Varroa mite. Varroa is, of course, very small. And so this is a scanning microscope picture just to show you sort of the details of the body on the back side, the dorsal side, and then the ventral side, how it looks like. And we can compare that to the Tropilelaps mite, which we, for good measure, also included in this study. And so Tropilelaps, as you can see here, is very different in body shape than Varroa. It has a very elongated body. And again, the back side here, or the dorsal side, is on the left-hand side, and then the belly side or the ventral side is on the right-hand side. You can also see that they share some commonalities with their legs being these typical ectoparasitic legs to hold on to their host organism. I should mention that Tropilelaps is not yet moved far out of Asia, but it seems to be on the move and expanding its range. And we are a little bit concerned that it might actually reach a global distribution just like Varroa. Now, depending on which countries or scientists in countries or beekeepers in different countries you speak to, Tropicolaps is much worse than Varroa, particularly seems to be the case in Thailand, or not that bad at all if you talk to, for example, my colleagues in China. But still, we have to be vigilant, and we don't want to keep it, want to keep it out. We don't want to keep it having to spread over the world. For us, it provided an interesting comparative data point because it feeds on honeybee brood and exclusively on honeybee brood, whereas Varroa, as we know, feeds on the adults and also on the brood, and on honeybee brood is sort of the reproductive life history stage. Yeah, so just to reiterate, so this was the study published by Samuel Ramsey and a lot of colleagues here in PNAS, telling us that our conventional wisdom that they're feeding the blood or the hemolymph of bees is not correct, but that it primarily consumes honeybee fat body, Varroa that is. But what the title doesn't mention, and we often just look at the title, is that the study was addressing the adult. And you can see this in this picture here on the bottom left there, where Varroa is wedged between the exoskeleton plates of the bee and sort of perfectly fits in there. It makes perfect sense that it sits there and it protects itself from grooming or biting of the bees and can then access the fat body that is just sort of the tissue underneath the exoskeleton. And that created, as you said, a lot of press, common press, but also was taken up by, government agencies such as the USDA ARS that then had on their website, Varroa mites feed on the fat body. And that's so far correct, but then they say, well, of adults and larvae. And that, is sort of our addition here to this study, that maybe on the larvae, on the pupae, fat body is not the primary food source. And they are going to change that. I've already talked to the people in Tucson. Anyway, so this is sort of the scenario that, yes, on the left-hand side here, and I want to give a big shout out to Alex Wild, who does fantastic nature photography. But on the left-hand side, basically, we have the adult host. On the right-hand side, we have the pupal host, and they're very different. And so on the left-hand side, in this slide, you see the feeding sites that are established on the adult, as Ramsey reported, and we used sort of the same technique, same method. We really tried to be in parallel to that study to establish where on the pupal host the Varroa mites are feeding. And you can see that on the right-hand side here, these colored circles are preferred feeding sites, and the darker the color, the more feeding there is on a particular site. And it matters because the fat body slowly only develops during pupation. And you can see that from here on the left-hand side, from the white-eyed pupal stage to the dark, almost ready to emerge stage down here on the bottom right, on the bottom left, sorry. This is a time series, and we dissected sort of this ventral side of the abdomen off and stained it for fat body. You can see the fat body stain here in red on the A3, B3, C3, and D3 panel. You can see that the red only very gradually appears and is present in significant amounts only at the very end of pupation. And so where the mites of Varroa is feeding for a large part of their life cycle doesn't have fat body present. And that was sort of the first indication that maybe fat body is not the main food source at the pupal feeding stage. We went then on to parallel the Ramsey study with double staining for hemolymph and fat body. This was a very smart idea to actually feed bees two stains that are staining in the living organism, and they're staining predominantly fat body in red, which is the color is called Nile red, and it's a lipophilic stain. And then on the left of that is a hydrophilic stain or a lipophobic stain that doesn't go into the fat but does stain the hemolymph or any aqueous substance, and therefore hemolymph on the second row here, you can see that the hemolymph is stained yellow or greenish, and on the third row, the fat body here is stained red. And so what they could show is that Varroa, after parasitizing the adult bees here on the bottom row, is predominantly stained red, which indicates fat body feeding. This is not, we didn't repeat that result, but we used that methodology to actually ask the same question for when we stain a pupae and then look at the VARROA mites. You can see that here in our slide. On the top row is the pupae. We have two pupae here. One is stained on the right-hand side, and the left one is just a control that hasn't been stained. You can see when we filter the light to red, you can see that the Nile red is in the pupae, and so is the uranin. The green is also in the pupae, and if you look at it combined, it kind of looks yellowish because green and red add up to kind of this yellowish tint. And then when we take a look at our protonymphs, deutonymphs, and adult varroa, you can see that after feeding on these pupae, they are stained a little bit red, but mostly green. And we can quantify that and look at sort of the intensity of these different colors. And this is shown in this graph. Basically, you have on the left-hand side the pupae. The pupae has a little bit more green than red. You can see that here. These two bars are a little different, okay? But when we compare this to the proto, deutero, or foundress mites, you can see the green is much, much higher than the red in all three of those stages. And I should point out that this is a logarithmic axis, so the differences are really quite dramatic. And this indicates that the different life history stages of the mite here during development, but also as an adult foundress, are predominantly feeding on the hemolymph, sorry, and this is a mistake in the title here, for the pupal hosts. So they are feeding on hemolymph, and predominantly so, and it's not due to excretion, because we also stain the feces, and the feces stain the very same way as the adult. We went then a step further and looked at proteins that are identifiable as honeybee proteins, so proteins that are in varroa and that are not completely digested, either because they haven't been digested or maybe that the varroa is never going to digest. But we can identify these proteins with molecular methods, and we can do this for the foundress mites here in orange, and then the dispersing mites in blue. And what we see is that there's a large number of proteins that we can find in both, but there are also sets of proteins that are unique to the foundress mite and the dispersal mite. Left is the foundress mite, and the right is the dispersing mites, and we can see that there's a lot more unique proteins that we only find in the foundress mites. There's some that are in the dispersing mites, but most important here is that we have, from a different study, from a different group, a set of honeybee proteins that have been identified in the hemolymph. The hemolymph proteins are indicated here in the darker color. And so what you can see is that the proteins that are unique to the foundress mites, there's about 3/4 of those are hemolymph identified or associated proteins, and only 1/4 is not. In the shared proteins that both foundress mites and dispersing mites have, it's about 50-50. And when we look at the proteins that are unique to the dispersing mites, there's actually 60 or 2/3 of the proteins are not hemolymph associated, or only 1/3 is hemolymph associated. So that tells us that the foundress mites are, first of all, they're different from the dispersing mites on the adults, and that they are feeding on different, presumably hemolymph proteins. We also looked at these overlapping proteins that are found in both the dispersing and the foundress mites, and found that most of those proteins were much more common in the foundress mites. So the foundress mites seem to be having a lot of proteins in their body from the bees, which makes sense because they need to produce the eggs and they need to be reproductive, which is mostly a protein metabolism. This includes major royal jelly proteins from the bees and also other storage proteins, so-called hexamerins. And in all cases, you can see that these proteins are more common in the foundress mite than in the dispersing mite. Now, we also looked at the varroa proteins, so not honeybee proteins, but proteins that are synthesized, made by the varroa, and looked at what kind of functions these proteins were indicating. You can see that these are numbers or words here on the left-hand side are a range of metabolic processes that are indicated in the foundress mites and the dispersing mites. but mostly we see a lot more in the founders mites than in the dispersing mites. And we can look at that in a different way here on the right-hand side. And again, there are things that are upregulated or overabundant in the founders, not so much in the dispersing mites. And they relate to a lot of different biological functions that are all telling us these founders mites are a lot more active and well equipped to be active through the hemolymph feeding. The other technique that we used is to look at not the proteins, but the little small molecules that make up the metabolism. And you can see here, again, the details are not that important, but you can see patterns of red and green. And I apologize for those people who have difficulties distinguishing red and green. The important part here is on the left-hand side where there is sort of this clustering, and these clusters are indicating similarities. And you can see the clusters are actually according to the food source and not the age of the individual. So we have a cluster here of the dispersing mites that is most different from all of the other clusters. So the dispersing mites are more different from the founders mites, even though they're the same age, and the founders mites are different from the protonymph or deuteronymph in their metabolism. And we associate that with those three life history stages feeding on the same host tissues, namely the hemolymph. We can look and analyze again sort of the dispersal and the founders mite stages and compare them with these small molecules or metabolic markers. What you can see here is again that in the founders mites there's a lot of upregulation. This dashed line here is sort of the line of significance, or the boundary line where we say this is telling us something or not. On the left-hand side we have 2 dots for the dispersing mites that are above that line. And on the founders mites, we have, about 18 or so. Again, indicating that there's a lot of metabolic processes that are upregulated in the founders mites. For good measure, we did look at tropilelapse and we found very similar results. Tropilelapse staining indicated that tropilelapse are also mostly hemolymph feeders because they are mostly stained in green. Again, this is sort of the actual data here where we quantified these stains, and you can see the pupae has a little bit more green than red, but the protonymph, the deuteronymph, and the founders of tropilelapse all have much higher green to red ratios, and the feces again has also a higher green to red ratio. When we look at the proteins for the tropilelapse, again, this is sort of this Venn diagram where we have overlap, and the green here on the bottom is the tropilelapse, are the tropilelapse proteins, and I should say the honeybee proteins that are found in tropilelapse. You can see that there are some that are only found in tropilelapse. So tropilelapse does have a little bit of a different nutritional niche, but there's a lot of overlap. And the overlap is exclusively with the VARROA founders or the shared proteins. There's no overlap between tropilelapse and VARROA dispersing mites. Again, telling us that there is this distinction of feeding habits between the mites that are feeding on the adults and mites that are feeding on the developing honeybee. And so, yeah, that's basically the conclusion that we conclude from this study, that VARROA feeds on the fat body of adult workers during the dispersal stage, as reported by Ramsey, but during the more important, I would argue, reproductive phase, they are feeding on hemolymph of the developing bees during their reproduction. We also show that hemolymph is a sufficient food source for them to upregulate their metabolism and reproduce. And there is another study from 2023 that also shows that hemolymph can sustain VARROA quite well. And then additionally, we show that tropilelapse also feeds on hemolymph. Overall, I think the take-home message here is that food choice is not just driven by nutritional value, but it's also an accessibility, quantity equation, and how much it costs to get to a food source, or whether you're going to be groomed off or not. And so nutritional value is not the whole thing. Animal behavior typically is a cost-benefit analysis, and the bee mites are no exception to that. And with that, I want to thank sort of my group here. That's sort of a snapshot of our group members on the left-hand side. again, Bin Han on the right-hand side there was the leader of this whole practical study, all the practical aspects. And we did have a lot of funders supporting us because science is expensive, and we couldn't have done it without them. And with that, I think, we can end up going into maybe a discussion of questions if your readers have some.
Thank you, Olav. Let me do some comments here, because one of the main challenges to control varroa mite is to understand the life cycle and his completeness. We need to know every single detail of the life cycle so we can try to find ways to intervene. And I think your research add a little bit more of information for all the researchers out there to understand better this life cycle, so we can maybe finally, after 40 years now in the United States at least, to find some way to control varroa mite.
Yeah,
that's all something important.
Yeah, I agree. I mean, we're all very worried about, this parasite that does a lot of harm to our bees. And, the existing tools that we have in our toolbox are going blunt, if you will. They're not the sharpest tools anymore. And so we need a multitude of approaches to find weak points in the armor or the life cycle of varroa. And, my lab is doing a lot of other research. So we are actually also developing varroacides to see whether we can chemically control varroa. I have a student who is looking for viruses. And viruses are, of course, another really big problem for beekeepers, viruses being able to attack and compromise the health of honeybees. But in this study, we are actually trying to find viruses that harm varroa instead of harming the bees as sort of a biocontrol method. And then along with Kara Wagner, who we also had on this show already, I know we're also looking at hygienic behavior and other natural defenses. And so this is, a multi-tiered approach that, yes, this study was about understanding varroa life cycle. This was not about a control method. But now that we know better and understand our enemy a little bit better, we can probably, come up with other ideas and new techniques or tactics to find ways to sustainably control it.
Yeah, I am looking forward to see what's going to come next from your lab about potential ways to help the bees against varroa, and also Tropilelaps. That is my prediction that unfortunately these things are moving so fast and people move bees all over the world. It's gonna arrive sooner or later. That's my prediction. I hope not.
Well,
I hope to be wrong.
We should all be vigilant. But, if you suspect as a beekeeper that you have Tropilelaps in your hive, it's really important to take high quality pictures with ideally with a macro lens or something like that, or collect them and send them to an expert, because there's a lot of little bugs that run around and that might look like Tropilelaps and they're not Tropilelaps. And they are difficult to distinguish if you just have sort of a blurry video or a shot. But, things that run around the comb very fast and are small and look, elongated should raise some eyebrows. And I agree, we need to be vigilant about it.
All right. Thank you for the message. That's very important. We need to be careful about this and send to the experts to a very wise identification. Otherwise become just words in the wind. I want to open the floor for the people at home. If you have some questions, now is the time to talk with Olav. And if you don't have any, I will be available in the academy this afternoon if you guys want to chat, if you have any other questions about anything else. So if you have some question, please write in the comment section and I'll read the question to Olav. Olav, Anne is asking, the Tropilelaps spread the same viruses as varroa do?
Yeah, that's a very good question. And I've just had 2 students in November and December in Thailand to sample bees with Tropilelaps and bees without Tropilelaps to address that question. We don't know at the moment what kind of viruses are in Tropilelaps, and it could be. My prediction is that there is a good amount of overlap because they are taking up the viruses from the bee hosts. But there could also be some unique stories that we just don't know about, and we have just collected the samples over the past few months and are processing those samples in the lab. as any other science, it's going to take some time because it's just not, very quick to get to the data. These are genomic techniques where we need to send the prepared samples to a center to get the data back, and that usually takes a few months. But we should have maybe next year, in 1 year, we should have that answer.
Fantastic. Anybody else have any question for Dr. Ruppell? All right. I think that's it for today. Olav, I want to thank you very much for not only for your work and for your time here today with us. That was a pleasure.
Yeah, it's my pleasure too. And have a good day.
All right. Bye-bye, everybody.